Hello everyone. This is Dr. Saurabh Kose, and an Associate Professor, Department of Pharmacy Practice, ISF College of Pharmacy, Moga, Punjab, India. Welcomes you all to this session on Introduction to Clinical Research. Clinical research is basically one of the scientific curriculum in which a specific place called Clinical Research Organization, the new drug development takes place, where pharmacological, toxicological, doses form designing, and various regulatory compliances takes place. An individual working in a clinical trial profession and doing clinical research has to undergo various regulatory subsets, standard operating procedures, and various kind of things which relates the successful completion of the phases. So clinical trial basically is divided into various steps. One by one, the things goes on. That is called as the phases of the clinical trial. So it is just a phase where one-to-one the things takes place. Coming on to the site where the clinical research has to be started. The clinical research is the one process which goes and followed by the previous one. So to start up within the clinical research segment part is the drug discovery process. And in this drug discovery process, we go with various kind of animal studies, lead compound optimization, pharmacophore, taking various molecular level, genetic level, pharmacokinetic, pharmacodynamic, all the genetic materials with a enzyme, tissue, a specific goal called target inside the body, where the mechanism of a molecule which is going to be approved is to work. And ultimately we develop this lead search, develop this microbiological assay with the diversity, and it's a very tiring process where the lead compound is optimized, its selectivity, its doses response is lethal dose at 50% concentration, effective dose at 50% concentration. Certainly, daily defined dose, maximum tolerated dose, overdose, underdose, missed dose for the profiling purpose of a molecule which is necessary. And certainly we do this all the things into the animal, rodent and rodent species comprising of one or alternate route of administration. And thereafter this microdosing phase finishes, we comes onto the phase 1 called as therapeutic exploratory phase, where the healthy volunteers in numbers are there, and in phase 2 called therapeutic exploratory, followed by the dose escalation phase where we titrate a safe dose, and this safe dose concerns with the compromising part coming onto the validation phase called phase 3, and into it therapeutic confirmations takes place. And thereafter we go on to the phase 4 post-marketing surveillance of the pharmacovigilance, where the individual case safety report of the individual patients in the market as well as during the clinical trials of phase 2 and phase 3, and periodic safety update report of a molecule takes place. In the same process in India, Pharmacovigilance Program of India has been introduced since 2010, where that the signal detection and various drug alerts are being issued. And ultimately, after the post-marketing surveillance, the molecule drug approval is given. And thereafter, also we go for further translational research called phase 5, where we change the route or a specific dose, or we go with the synergistic effect called fixed dose combinations, FDCs, etc. So target selection or the validation is one of the process where we have to see a molecule, specificity, genuineness. So we have selected a target, a molecular pathway, a disease, a research gap, existing need, novelty, safety, quality, efficacy, tolerability, mutagenicity, carcinogenicity, certain kind of hits, certain kind of hypothesis is generated. So illness or any disease, disorder, syndrome, any situation it may be which comes up, where we need to understand the molecular mechanism of the disease. We need to identify a therapeutic target in that pathway where the gene, it may be genetic material, it may be enzyme, it may be receptor, it may be ion channel, it may be a nuclear receptor, it may be of anything of the nature which comprises of. Then thereafter, we go with the nuclear receptor and demonstrate with the genomic material how a molecule is going to act exactly. When we talk about the drug discovery, so evaluation of the activity of compounds of the target, it may be in vivo or in vitro, inside the body or outside the body, where a lead compound called pharmacophore or new chemical entity has to be certainly taken up with the compound library, compound from the published literature. We have to see any kind of drug resource, either it is herbal, either it is natural, or either it is synthetic, semi-synthetic, or marine source, animal source. So thereafter, the structure-based design, rationality, rational drug design, and optimization to give a proof of concept that yes, the drug or the lead compound which we are going to forecast is a safer one. And thereafter, we need to give the drugs like property like pharmacokinetics, metabolism, off-target activity, safety assessment, preclinical, clinical, paraclinical, post-clinical dimensions of a molecule has to be undergo. So, drug development phase, when it comes on to the development part, right, starting from the preclinical to the clinical domain, we have to look after the certain processes where we go with the safety profiling. Various ICH guidelines are there. When we go with the efficacy guidelines, so GCP, Good Clinical Practices, which is E6, revised 1 and revised 2 guidelines are available for the standard reference one wants to pursue a clinical trial. So before initiating this preclinical phase, your clinical trial must be registered with the Clinical Trial Registry of India, CTRI, and the ethical clearance must be there, which is CDSCO and COMS approved ethical clearance of bioethics as per the bioethics and the biomedical ethics ICMR regulations. So the ethical clearance from the Institutional Ethics Committee or Human Ethics Committee should take place. Thereafter, you go in animal studies where we take a microdosing part of a dose. So that's why the phase 0, its nomenclature is microdosing phase. So microdosing phase is something where we take 1/100 part of a molecule which has to be given up into the animals. Rodent, we take one hand rodent, one hand non-rodent species of the laboratory animals. So this laboratory animal research and husbandry research takes place where an unknown compounds, where its toxicity, its safety profiling is not yet established, we are establishing for the human use or the clinical use and within the safety limit. So quality is another aspect, so that we go with various kind of unit operations, unit processes like safety study, toxicity study, drug metabolism, pharmacology, process R&D, chemical engineering R&D, manufacturing, bioprocess R&D, and compliances, formulation development, research and development, various kind of biological research and development, dose, and vulnerable populations, pediatric, geriatric, pregnant and lactating women, male, female, hepatic impairment, renal impairment, etc. And thereafter comes a community medicine department where the biostatisticians work with the data coordination, research information, clinical data management, and its various unit processes. Compliance with the regulatory authority part, which works under the project planning and management marketing. So in a nutshell, when a drug is within the animal studies, we apply for the investigational new drug application, whereas in the phase 1 we take some 20 to 100 volunteers, take drug for say 1 month, then the data entry is there. So what is the learning into it? What is the drug profiling, onset of action, duration of action, various pharmacokinetic, pharmacodynamic parameters in the healthy volunteers. And there should be one process which should take place as a biomedical ethics, is called as the informed consent process, whereas the person should be informed, she or he might be knowing that what they are taking up, and for that purpose voluntarily they are participating in this human trial. And certainly the observations comes up to the phase promotion after the phase 1 end point. So coming on to the phase 2, several disease volunteers are taken up. The information's effectiveness in treating diseases, short-term side effects in health impaired patients, any dose range, biological response, etc. So therapeutic explorations are done for to get the biological dose, which can be inert. In the phase 3 validation part or the confirmation, called therapeutic confirmatory phase, where we make a relationship between the establishment of the both processes, which is called as the risk ratio. So less common or longer side effects labeling information comes up, and the molecule is now at the post-marketing surveillance level or the pharmacovigilance level, which is concerned with the phase 4 of the trials. So the clinical trial continuously goes with the advisory committee, human ethics committee, infection control committee, and certain regulatory authorities like CDSCO, CIOMS, IPC, and various drug control authorities, human ethics committee, etc. Regulatory review team comes for audit, quality audit. So auditors, internal audit first takes within the clinical trial team, and afterwards the external auditor comes for the audit, and hence they have some reporting. And if anything is missing or need to fulfill the full process, then it becomes compliance, and that compliance leads to a abbreviated new drug application. This abbreviated new drug applications generally come up, and the regulatory authority check, forget everything, and then they allow regulatory authority to issue a license, activities, or a order for the marketing of a molecule approved. So worldwide marketing authorization, WMA, in 196 nations of the world approves the molecule. So as you see, in the drug discovery is a very tedious process where the various multidisciplinary activities like chemistry, intellectual property right, pharmacology, immunology, enzymology, and neurobehavioral activities, animal human research, and the safety profiling, and the dosage form designing, functional group, quantity structure activity relationship. In everything in the drugs, in a nutshell, for preparing a drug, you see the pharmaceutics person, pharmacology person, pharmacy practice person, quality assurance person, bioengineers, and toxicologist, and certainly chemist. Everyone is involved, till regulatory authority, quality assurance. So you see a drug discovery process comes also from the natural resources. So pharmacognosist or this phytoconstituents drugs are also available. So everywhere the molecule is going to affect the anatomy and the physiology of body, human body, right? So its safety profiling is a one must criteria to be taken up. So it directly affects. So that is what a drug is called as. If any drug is into that profile, into that defined dose, it is safe. It is overdose, then it become toxic and it become poison. So that is why the molecule redefining and learning from the past unsafe human experimentations, such as Nazism experiment, or thalidomide disaster, or coming up with the various past unsafe human experimentations, which shows the timeline of the clinical trial and states us to do the drug discovery part in a proper process. So thank you so much for joining into this session of clinical research. Thank you so much.